afternoon, and thank you for joining us here today. We are here representing the Florida Institute of Technology with our project exploring the impacts of microgravity on ciliary and flagellar development. My name is Lucy Turner.
And my name is Trevor Mello.
And our other teammates, Josh Ahrens and McKenna Taylor, unfortunately could not join us here today. And we would like to say a special thank you to our teacher facilitator, Dr. Andrew Palmer. So what lies ahead for humanity is extraterrestrial settlements on the moon, on Mars, and potentially even other planets outside of our solar system. However, currently, humanity's extraterrestrial habitation is restrained to more temporary structures such as the International Space Station. However, life on the space station and travel to these other planets both have something in common, which is long-term exposure to microgravity. Therefore, it's important to understand the effects of microgravity on the human body if we want to establish successful settlements. And I'm talking about effects such as on things like flagella and cilia. Now, these are motility organelles found in some cells. They are similar, very, very similar in structure, although there are some differences. However, you can study cilia and apply it to flagella and vice versa. Now, on the right of this slide, you will see a diagram showing where in the human body cilia and flagella are present. It's important to note that they are involved in crucial life-sustaining processes such as maintenance of the brain, breathing, and even reproduction. So these are very important systems that are involved in the survival and propagation of our species.
And the organism that we chose to use is everybody's favorite eukaryote, algae. Specifically, Chlamydomonas reinhardtii. Now, there's 3 specific reasons why exactly we're using Chlamydomonas reinhardtii. Number 1, it is genetically and biochemically tractable, which simply refers to the fact of how easy it is to manipulate the genome and also to manipulate the biochemical processes and to further understand the biochemical processes that are at play for this organism. The second is it's easy to grow. Depending on the size of the culture, whether you have 1 mil or a liter, it is very, very easy to get it to an exponential growth rate. And we've shown in our own lab that we can get this exponential growth rate in a 15 milliliter culture in a couple days, which is relatively quick. The last reason is that it is very inexpensive. The media that we use is tris acetate phosphate, or TAP for short. And it is very inexpensive, and it's not that you need a lot of it per se. It's more of the fact that the organism itself is very opportunistic. Now, the big question that this experiment is trying to answer is what does the effect that microgravity has on flagella and cilia? Now, there's been a lot of talk about microgravity and simulated microgravity. Simulated microgravity is not regular microgravity. Simulated microgravity simply disrupts the gravity sensing mechanism, excuse me. Now, this experiment here, if I can get the pointer to work the right way. Okay, maybe not. So for the figure A here, we see that normal gravity is in the top, simulated microgravity is in the bottom. This is an immunofluorescence assay, and the primary cilia is stained green. Now, as you can see, over a 24-hour period, the normal gravity relatively stays the same. You see a little bit of changes, but the simulated microgravity, however, you see a drastic drop in the amount of primary cilia. In part B, you can see that the cells with primary cilium, is a lot less than the normal gravity when compared to the simulated microgravity. Same thing for part C. You can see that not only is there less cells with primary cilium when simulated microgravity, you can also see that there is a shortening of the length of the primary cilia. In our experimental design, we will have TAP agar that has a live culture of the Chlamydomonas. We have regular liquid TAP, which is the simple tris acetate phosphate I was talking about earlier. The clamp will be opened at U-14, shaken gently, and the glutaraldehyde acts as a preservative and also kills the algae to ensure that the effects of Earth's gravity doesn't have any effect on the ending result.
So to test our proposed experiment, we grew algae in a stab culture. And I'm just going to breeze over this really quick for the sake of time. But essentially, we found that our proposed methods, the algae would survive through the flight on the throughout the course of the experiment, which is important until the moment that we kill it with glutaraldehyde and preserve it. And that's essentially what this data is showing. And then on the bottom right, you see a picture of a phase contrast microscopy, which is what we would use to analyze the data once it's returned to us back to Earth and measure the flagella. And thank you very much.
Great job. Do we have any questions for this team? Oh, one question over here.
Like innate, and then as soon as it gets into the media, is it being activated, or is it alive the whole flight?
Yeah, that's a great question. Thank you. So we were planning to send live culture because Chlamydia monocyte is fairly resilient, and being stabbed in the auger, it would offer some protection, but the algae can survive for an extended period of time just in a culture like that. And then the liquid tap that we would be introducing in the first clamp, if you remember the diagram, would be to get the algae into the liquid and swimming and growing their flagella in the microgravity conditions.